Hello and welcome back to Science News Today. I am Nathan Vanderveer Harris,
I'm Philip Kearns.
And I'm Celia.
and we are here with a very fascinating segment for you today. We are going to be discussing a very interesting and recent study comparing Mars as an analog with Yellowstone National Park and the microbiology involved at Yellowstone and how it relates to life on Mars.
So what we're specifically looking at in this study is comparing the different types of microorganisms, specifically the bacteria that have been discovered in different areas of Yellowstone in the hot springs. And we're going to look at the development of these organisms and their habitats, and then try and compare these with the potential biomes and the development of similar organisms on the surface of Mars.
This study is led up by Dr. Sherry Cady, who is a professor at Portland State University and the editor-in-chief of Astrobiology magazine. Astrobiology, as we're referencing it here, is the science of life in space.
Yellowstone's a great place for getting a handle on what it takes to actually have microbial life enter the geological record, and what it might be like for those microbes that live in extreme ecosystems, what we consider now today to be extreme, but on early Earth were not as extreme as they are now. These organisms occupy the hot springs from the lowermost temperature way out at the distal ends of the systems up to the near boiling temperatures at the hottest part of the pools. And so we can really get a feeling for how these organisms become fossilized, how they enter the geological record, what kinds of traces they leave behind. And all that information helps us better recognize signs of life in the geological record, whether that geological record be on ancient Earth rocks, or whether it be looking at rocks on another planet like Mars, another rocky planet like Mars.
So these fossils of the microorganisms are actually the early evidence that we have of life on Earth. And the oldest fossils among these are roughly 3.5 billion years old, which is actually only 800 million years after the Earth first formed. And to understand really how the first living cells appeared, experiments have been conducted in geology, chemistry, biology, and physics. And these have actually led scientists to hypothesize that the chemical and physical processes on early Earth and the atmosphere there could have produced simple cells through four main stages.
These four stages begin with the non-living or abiotic synthesis of small organic molecules such as amino acids and nucleotides. From this stage, the joining of these small molecules into macromolecules, including proteins and nucleic acid, takes place. After that, these molecules are packaged into protobionts, droplets with membranes that maintained an internal chemistry different from that of their surroundings. Lastly, the origin of self-replicating molecules eventually made inheritance possible.
And the great thing about Yellowstone is that we can actually see how these four stages might have come to be in conditions that were more similar to early Earth. And the first atmosphere, as we are aware, was probably thick with water vapor, and there were various compounds that were released by these volcanic eruptions, including nitrogen and its oxides, carbon dioxide, methane, ammonia, hydrogen, and hydrogen sulfide. And what's great is that we can see all of these compounds in the hot springs at Yellowstone.
And so evidence that these compounds were really conducive to early life on Earth can be seen in the hot springs, where you have these organisms that are incredibly robust, standing temperatures of over 140 degrees Fahrenheit. And in addition to this, they can rapidly colonize essentially anything that they come into contact with.
In a study conducted by Mike Rodriguez at Yellowstone, we can see the colonization of these microorganisms on slides that were placed in hot springs in Yellowstone.
We set some glass microscope slides in the spring overnight in the hottest part of the spring, and I was just amazed at what we saw when we looked at what colonized that slide in a 24-hour period. When we looked at it through the microscope, there were parts of it that had a very dense, complex mesh of microbial growth on the surface of the slide. So it was really surprising to see how quickly the microorganisms colonize just a blank glass plate, and how quickly they divided and how quickly that colony grew.
Hey, thank you so much, Mr. Rodriguez. That's quite insightful. And what really intrigued us here at Science News today was the fact that these organisms, these microorganisms, could in fact thrive and live in such distinctive temperatures and environments. After all, we learned earlier this year about the importance of homeostasis on organisms and cells, and so we were just blown away that they could live in such extreme temperature.
To get a better idea of exactly what these microorganisms were going through, we actually had the opportunity to talk to Secret Ballinger about the conditions that they were observing these microorganisms in.
That's right, and Secret Ballinger was also present with Dr. Katie in her study at Yellowstone.
Okay, so our initial hypothesis was that temperature and or pH might have something to do with the distribution of those bands we saw along the outflow channel. So we took temperature readings and we took pH measurements, and we found that actually the pH was the same pretty much throughout the channel. And then when we took temperature readings, however, we did find a difference. We found the further edges of the channel were lower temperature. The center of the channel, where the flow was fastest and where it was deepest, it was hottest. And in the middle, we were only finding the cream colored at that deepest, hottest part, whereas we were finding yellow bands out towards the middle, and then towards the edges only orange bands. We seem to have found evidence to support our hypothesis.
Thank you so much, Mrs. Ballinger. Now, Phil, I know that prior to this recording, you've done quite a bit of research on how these thermophiles are able to withstand such high temperatures. Is that correct?
Yeah, that's right. And actually, hypothermophiles, just a little bit of background, they were first discovered in Yellowstone. And since then, more than 70 species have been discovered. But the two things that are really key to the success of these hypothermophiles is that their protein molecules exhibit hyperthermostability, which means that they can maintain their structural stability at exceedingly high heats, which really no other organism can. And the cell membrane that encompasses them contains really high levels of saturated fatty acids, which helps them to maintain their shape at high temperatures, thus allowing them to function properly.
Now, Celia, you, I know, did some research on the other side of things that Mrs. Ballinger was talking about, the pH. Is that correct?
pH homeostasis is yet another way that microorganisms are able to maintain stability in their environments. The level of acidity in a substance is measured through its pH. And although most microorganism species require external pH levels near a neutral level, which is a pH of 7, bacteria called extremophiles can live in environments with a pH value as low as 3, which is incredibly acidic, or above 11, which is incredibly basic. Bacteria have mechanisms for sensing external changes in pH, and this allows these microorganisms to enable them to tolerate external pH values that are different to their internal levels of acidity.
Now, Celia, what can you tell me specifically about how these extremophiles handle the varying pHs?
One particular extremophile that is really interesting are called acidophiles, which are the microorganisms that I mentioned before that can live in pHs lower than 3 or incredibly acidic conditions. These acidophile organisms have evolved extremely efficient mechanisms to pump protons out of the intercellular space in order to keep the cytoplasm at or near a neutral pH value of 7. Therefore, intracellular proteins do not need to develop acid stability through evolution.
So now that we've spent so much time looking at the micro side of things, we thought it'd be interesting to zoom out and actually examine the populations of macroinvertebrates that we have here in Yellowstone. And these are primarily found in the pools of recently formed hydrothermal precipitates that we have in the springs.
And to supplement our research of the macroinvertebrates at Yellowstone, we have here with us today Miles Hudson and Stephanie Pringle, who work together at one of the thermal vents at Yellowstone. Miles, what can you tell us about what you guys studied at Yellowstone?
Initial observations were first geared towards macro invertebrates, larger invertebrates. But after talking with Dr. Katie and sitting in front of the microscope, I definitely personally kind of focused more into the invertebrates that were surviving or living in the bacterial mats, the biofacies on the lower temperature gradient area.
Okay, and Stephanie, what can you tell us about the macroinvertebrates that you found in those lower temperature gradients?
We found and described about a dozen or so macroinvertebrates, from tiny nematodes to beetles to stonefly nymphs, up to the larger macros, to dragonflies and damselflies that we saw flying around. So it was very interesting to see that they've adapted to this warm water and alkaline water.
Thank you so much, Mrs. Pringle. Now, we've been able to see that there is both life in microinvertebrates and macroinvertebrates at Yellowstone.
And so, basically, to wrap this all up, what we can see is that the large biodiversity in terms of both micro and macro invertebrates shows that with the conditions observable here and potentially observable on Mars, especially in the early stages of the solar system, there is a high ability for these organisms to kind of develop, and this really lends us insight as to what intelligent life or even just cellular life might look like on other planets.
On behalf of Philip Kearns, Nathan Vanderveer Harris, and myself, we would like to thank all of our listeners out there for joining us today on this segment about the microorganisms and other forms of life at Yellowstone National Park Hot Springs. Please join us next week as we discuss another topic on Science News Today.